All right, so last topic for the module 3, and it's fairly easy, but you will see that I have written in some of the slides that take a look at the experiments, because we are going to ask a couple of critical thinking questions. You will see them in your quiz as well. We will comfortably finish this one because the material is easy. I do not need to spend too much time on each slide. Many of them are self-explanatory, and you can just read and understand that it's not complicated slide at all. So just to say that we have met the enemy and he is us. Anybody knows that where the code is from? It is not the original code. It is so. It was that we have met the enemy and ours was the original Commodore Perry. That was the Battle of Lake Erie that happened in 1812. That was the original in Put-in Bay in Lake Erie in Northeast Ohio. That was the original. And then in 1965, it was the first Earth Day that was celebrated, and there was a cartoonist named Walt Kelly. He actually wrote that, and basically what they're seeing that in front of them, it is just trash and garbage, basically. So you understand that is like basically we are polluting our own environment, and that's the enemy of us. And here, why it is relevant that you're going to see that in many of the viruses, we already know that some of them are cytopathic. That means they are going to kill the cell directly by many different means. But a majority of the viruses may not be cytopathic, but it's us who kills or cause the disease, which we call as an immunopathology. The virulent proteins of the viruses, or even some of the sequence of their genome, like the DNA or the RNA, that cause this immunopathology, that our immune system is overly reacting, and that cause many different problems, and we are going to see that. So just read the blurb of the opening slide. It will help you understanding that. So here, some basic definitions. Pathogenesis is the capacity of the pathogen to cause a disease in a host. Most of you are microbiology or biological sciences. You will know this. And then it shows signs and symptoms. And symptoms, what you can feel like a headache or like a body ache, those are the symptoms. And signs, what can others detect? Say, for an example, the rashes or a fever or any outcome of the disease that you can see. Reddening of the eyes due to vasodilations and those kind of things, those are the signs. Virulence, on the other hand, is the severity or the degree of the pathogenesis. I mean, like how bad it is versus not that bad. Just think about if you just think about 2 different episodes of cold, you just always do that. But again, for the viruses, we have to be very careful about that. And it can be measured or quantitated, but it is a little different than many other microbiological agents of the pathogen, and we'll say that why. Viruses can be virulent, avirulent, and attenuated. You already know what virulent means. Avirulent means, like, say, for example, insect viruses or plant viruses. They pass through us, avirulent. And attenuated, mostly, they are naturally occur in our body as attenuated as they are mutated. We just never know that. But along with that, those attenuated many strains which are developed in the laboratory can be used as a vaccine as well, that we're going to talk about. So how we quantify the virulence? We can compare them with the incubation time, virus titer, mean time of death, fever, weight loss, pathological lesions like poliovirus, like in the brain or anywhere, like these are in the experimental animals, and say reduction in the blood cells. So here, just remember that your CD4 cells are the T helper cells, and the CD8 cells that we are going to talk about are the cytotoxic T cells. So the CD4 T cells are to which the antigen is presented, and it alerts by the Th1 or the Th2, T helper 1, T helper 2, 2 lineages of the CD4 T cells, which alert the immune system. So just keeping back in your mind that we're going to talk about the CD4 T helper cells many times, and it will have 2 lineages, CD4 Th1 and the CD4 Th2, producing 2 different types of cytokines. We're going to come to that. So that's one of the things. And here, just a couple of incubation periods. You do not have to memorize anything. Just shows, just from, say, influenza rhinovirus, 1 to 3 days or 2 days. In some of the cases, like particularly hepatitis viruses, rabies, papilloma, it could be many months before the onset of the disease. Sometimes you cannot just even see those diseases. These are some of the interesting incubation periods that you can see, and we're going to see that why it is. So that's about it. Now, how we actually study the viral pathogenesis and what are the limitations. So the biggest problem that what we have, and that's the reason that there are so many hundreds and hundreds of papers are published. Okay, all right, my antiviral works, this medicine works, that medicine works, all works in cell culture, but there is none in the market to cause this. So it is easy to publish a paper out of a cell culture, but hardly anything ends up in the market. The difference is that the cell culture doesn't have an immune system. So it is no way you can compare an animal in vivo what is happening. Not only that what happens in the mice is not going to happen in the human beings as well. So the first thing that this very interesting experiment that I'm just describing, just one of them. There are many of them you can just find. So the human cells, it was already understood that the ICAM receptors are the basically is a intercellular adhesion molecules basically acts as a receptor for the influenza virus. And on the top of the ICAM, at the very end, you have a sialic acid. Sialic acid is N-acetyl glucosamine or NANA. So you have a sialic acid. The sialic acid is the sugar which is sticking out at the very top. That first sugar is connected to the second one. In the second position, there is a galactose. And this galactose is connected by the carbon alpha-2,6. Alpha carbon 1 of the sialic acid is bonded to the alpha carbon 6, alpha carbon 2 with the alpha carbon 6 of the galactose. So that's an alpha-2,6 linkage. That is in the human being, and it was thought that that is what is required for the influenza virus, and it uses that receptor. It's proven in the cell culture. That is what it is. So it is coded by a sialyl transferase in a gene which is called ST6-gal or STGAL6. ST6-gal, both ways is correct, is coded that sialyl transferase to make that linkage. So what it has been done, the scientists actually took the mice and they knocked out that gene, which produces the sialyl transferase to produce that alpha-2,6 linkage, challenge them with the influenza virus, it infects the mice. So what was thought before is completely that idea was not there anymore, and they found that that the alpha-2,3 linkage. So here, the first sialic acid or the N-acetyl glucosamine, same thing, is connected with the galactose instead of an alpha-2,6 with an alpha-2,3 linkage. So these are the things these are the limitations of the discrepancy that the cell culture doesn't tell us what happens in vivo in an animal model. And the next is that this is also another funny code, but the mice lie and the monkeys exaggerate. There are many of these disease models that we start in these animals. Because you think about that, you cannot study a viral disease in in a human model. Although there are some examples of a rhinovirus as well as influenza virus infection where there were volunteers, you have to pay them huge amount of compensation. Remember that it is not a vaccine trial. This is a disease trial. Somebody is giving you a virus that will cause a disease. Even if it's common cold rhinovirus, there are implications, and it is not feasible, it's not done. This is not the normal way. So you have to have animal models, monkey models, and many times the results are different. So what are the different ways that we can do these experiments? So here, what some of the examples are the mice synthesizing human receptors. Transgenic is a routine development. These are, you do not have to develop in the lab. You just buy them commercially from the companies. They just have these human receptors, mice carrying complete viral genomes. Say, for example, it was very difficult to grow the hepatitis C virus in the hepatocytes or in the liver cell. It would just not replicate. It's very difficult. And what you can do that you can clone the entire viral genome into the mouse, and these viruses will produce the viral proteins, if not full virus particle, in the mouse. And you can just look at the pathogenicity and the virulence, everything else. And also, what you can do that you can express viral genes. You can just think about that, okay, all right, this is number 1, this is the X gene, this is a virulent gene, you clone that one, and if it expresses, that can show that whether that virulence gene is not there or not. So if you have the gene, you have the virulence. If you knock out the gene, you don't have the virulence. These can be determined. And then similarly, there are many different things what you can do. You can just put the T cell receptors. Then you can just immune mediator deletion. So you can just have a mouse where you can just delete a TLR. Say, for example, a TLR which detects something like specific. You can detect that. And if you challenge it with the virus, see that what happens. You just can delete the B cells, or you can just delete the T cells. These are mice routinely available, which doesn't have any B cell or the T cell, or even both. These are called SCID mice or nude mice. SCID, SCID, so severe combined immunodeficient mice. They have no FAR in there. They're called the nude mice. They're just like pink skin in there. Probably have seen pictures of those. Doesn't have any T cells or the B cells. These are the models for many viruses which can cause cancers, except or other things. Immune mediator overproduction, very important. If you just produce too much cytokines, a particular one, or in combination, what happens? What kind of immunopathology happens? These can be studied by these processes. So this is basically the ways, although there are limitations, but still significantly there are some advances out there. So after that, we are looking at some of the pattern of the infections by the different pathogenic viruses. The first one, we are looking at an acute infection. Usually it's just like a quick onset of the symptom, or after the incubation period, there is initial vague symptom, the disease peaks, and you have a convulsant period. And a typical cold infection, say rotavirus, influenza virus, we have seen all these cause this acute infection. Then what you have is the latent infection. The latent infection is a typical herpes simplex, say for example, the cold sores. The virus produces the disease, and then there is no more virus production. There is a latent period where the virus completely disappears. You cannot detect the virus. It just resides in some cell and don't replicate. Just stay in there in a latent form. And usually this is how it happens. We talked about that before. Their DNA is methylated, and there is these CpG methylation to silence them, and their genomes stay in there in a silent condition. And sometimes they can just come back. You can have a virus production in which you can have the onset of the disease, or you may have a virus production which is the virus is detectable, but you may not see the disease, asymptomatic viral reproduction. Any combinations can happen. On the other hand, here we are looking at this persistent infection by this lymphocytic choriomeningitis virus, or this John Cunningham virus. You actually, once infected, produce these viruses in a small amount for the rest of your life. The virus is produced in a small amount, and usually this is in kind of cells or the tissues where there is less immunosurveillance. So those type of tissues can replicate this virus or shed the virus for the rest of your life. But you see that there is no red bar. That means that it can be asymptomatic. Whereas here, what you are seeing in the persistent infection, once you get infected, these persistent infections can stay there for a very long time, or even sometimes life. Say, for example, for the retroviruses like immunodeficiency T cell lymphotype, that's for your life. Measles virus, for a long time, it can be cleared. But what happens is that sometimes you have an acute infection at the initial period, and then you have a very low level of a virus that is being replicated, almost undetectable, sometimes detectable, and then there could be an onset of the disease which can cause even death. Not always, but that can cause even death. So these are the different types of pathogenic virus comparison. So here we are looking at a couple of models that were very interesting, and the reason is that this will cause, or the same virus will cause an acute infection, and it will become a persistent infection. That's why we are looking at this. So here, chickenpox virus, which is a varicella zoster, remember that it is a herpes virus. So you initially have about 4 to 6 days. You will start the primary viremia, reproduction in liver, spleen, other organs. Then from there, the secondary viremia, it will go into the infection of the skin. And about 2 weeks after the initial exposure, you will have the onset of the rash. And then what happens is the most important part. So as you have that all over your skin, all over your body, you have sensory nerve endings. These are your peripheral nervous system. And these cells that what you are seeing right here, this is right here is a sensory ganglia where you have the cell body. And these are the axons. Remember that this is just only one axon. This end is also an axon. And it just delivers the axon or the action potential to the central nervous system. So these are just with just one axon. And this nerve ending, which is basically many types. This could be touch receptor, mechanoreceptor, thermal receptor, all these receptors throughout your body. And sometimes they are modified, this ending. The virus enters through here, and then it just infects or basically comes into the body of the neuron. These are your sensory neuron in the ganglia. If you have chickenpox, you will have, or anyone who has chickenpox will have these latent viruses in their sensory ganglia somewhere. There is no way that anyone can, there is no exception. They will be there. Now, for some, nothing will happen. And let's see what happens to some other people as well. After many years, which is, you already know that reactivation can cause the shingles. And this may happen due to sometimes stress and other onset of other physiological conditions. But again, it can happen in some, it cannot happen to many. There is a good vaccine against that. And also the important part is that what is seen that when you have the shingles rash, unlike the initial chickenpox that can have over the body, in the shingles, you will have just only a patch of your skin, which could be face, body, back, anywhere. Just a patch of your skin is basically affected, along with the severe pain. The reason is that each of these areas of your body, which is innervated by these peripheral neurons, they are called the dermatomes. So, for an example, if I'm just showing that here, all of them are showing the same thing. If the virus is in this first neuron, it will affect usually the nerve endings, which is going to a very particular, say, region 1 of your skin, which could be the face, anywhere where it is innervated. Instead of this one, if it is in the region 2, it will just appear, the shingles will appear in the dermatome 2 or the dermatome 3. So that's why it's very localized as such. And it's much more painful compared to the initial infection. So here it is that how the herpes simplex, particularly, say for an example, just have latency. And this is just not your varicella zoster, but this is just like all other herpes simplex viruses or other herpes viruses. They can just gain access through the skin, and then they go through into the dermis. And from there, they can just get into the axon of the sensory ganglion. And they just go through the long axon and then harbor into the sensory ganglia for the rest of their life. On the other hand, just this is from your physiology. I'm not going into that. There could be another thing that say you have blood vessels in this dermis area, sweat glands, and these blood vessels, sweat glands will have autonomic nervous system innervation because, dilation and constriction of these blood vessels, these are controlled by the autonomic nervous system. So this part is the autonomic nervous system. You have this sensory sympathetic neuronal cell body. This is the axon for that, where it goes. It can infect your sympathetic neuron cell body as well. So the first one, this is your peripheral nervous system. The second one, this is your autonomic nervous system. Both of them are synapsed or connected with the central nervous system as well. It is rare, but sometimes in some of the very unfortunate cases, these viruses can infect the central nervous system as well. These are very rare, but if it happens many times, this could be fatal. So that's the sad part of it, that it is not shown, but there is always a possibility of that. All right, so here is another example of a primary and persistent infection, and here it is Epstein-Barr virus, and this causes the infectious mononucleosis, or the mono. This is also a herpes virus, same thing, like just a different group basically. But the herpes virus can also go through the skin, and they infect the resting B cells. Once these resting B cells are infected, what they do that they will produce a large number of different proteins. These are called the eBNAs, or Epstein-Barr virus nuclear antigens. These are all in your notes. No worries with that. And also, they produce these two types of protein on their membrane, what they call as an LMP1 and LMP2. These are latent membrane protein 1, latent membrane protein 2. These are produced. When these markers are produced, at that time, the cytotoxic T cells, natural key cells, they recognize that these B cells are basically infected. These are called the B cell EBV infected, B cell classed, and the cytotoxic T cell or natural killer cells, they will kill these B cells. So that's the normal thing, and that will contain the disease. But this is the important part, that 1 in 100,000 on an average, 1 in 100,000 of the B cells will survive. And now we are looking at the second part right here. So the same thing, these are resting B cells, or these are the lytic B cells, which will kill the Lee cells, but some of them right here will become latent, and these will not produce this latent membrane protein 1, but they just produce this LMP2 or the LMP2A, you will see that, mRNAs, and they just produce these. So they can produce some nuclear antigens, or they may not. But these becomes basically the latently infected resting memory cells, and they can persist throughout the life of a person. And as sometimes these latently infected B cells can get activated, and the activated ones will also produce the LMP1. So think about this, that if the LMP1 or the latent membrane protein is produced, usually those cells will be recognized and the cytotoxic T cell will kill them. But if they don't produce the LMP1, they are usually in the latent stage, and they will not be killed by the cytotoxic T cells or the natural killer cells. So this is how in the B cells, the Epstein-Barr virus rest and cause this infection. All right, so viral virulence is a relative property. So one of the things that this is that what you have seen in the microbiology, we've many times compared the disease. And usually when you compare the disease and we just told that, okay, this is a more virulent virus, this is a less virulent virus, that's from an epidemiological standpoint. Remember that. It's an epidemiological standpoint, not at the molecular or microbiological standpoint, because you really cannot compare virus, which seems very different. I mean, like if you just tell, okay, all right, I cannot compare the virulence and cannot say Ebola or the rhinovirus or the influenza or the Ebola virus, whether their virulence is less or more, I cannot tell. So, so people will say that you are crazy. Like, you can tell it epidemiologically, but not at the level of the molecular biology or at the pathology level. And this is why it is. So what we are looking at, see, this is right here. It's all written in your notes. There are two different types of the poliovirus, type 1 and a type 2. And you can easily see that both of them initially, this is like the number of survivors and the day of post-infection, that how the different type 2 and the type 1 poliovirus, they have the difference in their virulence. Not only that, here you can also looking at that this is the different viruses. So you can say the Japanese encephalitis, yellow fever, West Nile, and each of them is different portion of the brain stem. So the right here is a cerebrum, and then you have the brain stem, and then you have the spinal cord. So these are the different region of the central nervous system, and you can easily see that how they differ where the inoculation is. So this Langat virus, this is like replicates the most right here. When it is the route of inoculation is the spinal cord. On the other hand, for these, the spinal cord is kind of okay. Here it is. You can see that brainstem is the main thing. Here, cerebrum, brainstem, both are good. Spinal cord is a little bit less. So these are some of the pathological lesions that differs, and that's the reason that when you are measuring the viral virulence, the virologist always will tell that you have to tell the virulence in the way for one virus. If it's a poliovirus in the first case, type 1, type 2, you can measure the virulence. If you are talking about different viruses going into a particular route of infection, you can compare Japanese encephalitis with yellow fever and compare that if you give both of them at the central nervous system, you can compare them. But if you give one virus naturally to the central nervous system, another goes through the oral route, and that you cannot make those comparisons. That's what I meant to say. All right, so here is also another example of that, what I exactly said. So here, this is the lymphocytic choriomeningitis virus, LCMV. It's a mouse virus. And if you just, in the peritoneum, intraperitoneum, if you just go give them about 100,000 PFU, the outcome, the mice survives. But just one PFU, look at that, intracranial in the brain, if you just give one PFU, it causes the death. And the cause of the death is basically immunopathological, basically. And the immunodeficient mice, on the other hand, survives persistent infection. What does it mean? That suppose, say that just one PFU, the mice is dead. This is basically the brain cells are attacked by the immune cells, microglia and other cells. On the other hand, what happens or even the other proteases, TNF alphas and all those, which causes the tissue damage, destruction, and lesions, those basically are the pathological outcome. But if the mouse doesn't have an immune system, and if you just inject the mice with the virus, there is no immune mediator, and at that time, the mice will survive. So this is what we call as an immunopathology, that your own body's good to clear the virus is basically doing the killing. And we're going to talk about that a long time. So basically, the goal of virology to identify genes that determines virulence, and usually identified by the mutation, a virus that causes reduced or no disease in a specified system, basically. So this is very self-explanatory. That's basically the goal. And how you are doing this. This is just like a very general scheme of that, and just you will see that there are several slides that I put carefully understand the rationale of the experiment. So here what you are looking at, there are two things what you are looking at. You are taking the wild type virus, you are growing it in culture, in the cell culture, it grows great. Then say for this particular virus, it is a neurovirulent virus, and you inject them into the brain and you see neurovirulence. That's the common outcome. Then you mutate different genes, one gene, multiple gene, whatever it is. And in that case, what you are seeing that it is growing in the cell culture very poorly. You inject that same virus to the brain of the mouse, and you can see that there is not much neurovirulence. It is attenuated. In this case, you cannot conclude that you have found the neurovirulent gene. See that here in the second case, the virus reproduction or the replication of the virus is overly minimized or is drastically reduced. That means the number of viruses are less. That does not mean that you have found the neurovirulent gene. This is the most important part. The virus doesn't replicate well, so that's why it's not causing the disease. The third one is the one which is important, that here you did a mutation in a gene specifically required for virulence, and you can see right here that it grows perfectly in cell culture, just like the wild type. Now, if you inject that to the mice brain, you do not see any neurovirulence. This is attenuated. If you have this case that it grows perfectly in cell culture, but not in the animal, this is your attenuated strain, and you have found your gene which is responsible for the virulence, not the second one. So just understand that we can just have some questions, critical thinking questions on these experiments, what to conclude. All right, so the viral virulence genes, they are mainly 4 categories. These are the viral virulence genes. The first one is basically has to do with viral replication, which is, you can be subdivided that invasiveness, that is like which are the tissues that it will go, and the tropism, which are the tissues. Invasiveness is like how infectious it is or how invasive it is, and the tropism is that in which other tissues or the cells that it will replicate. Some of them modify the host defense mechanism. We have seen a large number of this, enable the virus to spread to the host, and how it does that in many, many different ways. We have seen those as well. And the intrinsic cell killing effect, these are basically by production of toxins directly to kill the cells. So let's take a look at a couple of these examples of the virulence genes. So first of all, that the viral virulence determinants need not encode proteins. So this is the most important part, that what you are seeing, that sometimes the virulence is associated into the untranslated part of the genome, and this is true for the poliovirus. What we are looking at, that this portion that is shown in that inset, that is basically the 5' untranslated region of the poliovirus genome RNA. So that's what you are looking at. And the AUG is the first start codon, and all of these happening is before the first start codon, or before the coding region. The blue is the coding region. This is the untranslated region, and you know that this basically works as the iris or the internal ribosomal entry site. And what we are looking at is just this 5th secondary structure, this 5th stem loop right here, and this bulge out, what you are seeing, we are looking at this structure. You do not have to memorize that, but just showing that this is in the 5' untranslated region. These are the Sabin vaccine strains. These are the live polio vaccine strains that is used, and there are the mixture of these 3 different ones, type 1, type 2, and type 3. Each one of them has a mutation, and that make these vaccine strains attenuated. Remember that. Just that point mutation make these strains attenuated, and what they are. For P1, you have a 480 A to G mutation. Then in the P2, 481 A to G mutation. And in the 5' UTR in 472, you have a C to U mutation. So these single mutations in the 5' UTR can change the virus into an attenuated strain. It's like really amazing. No proteins, just the genome RNA. On the other hand, what we are seeing that the P3 Sabin, that can also cause something which we call as a reversion. This reversion does not mean that the 472 C to U will change back to C. Not that. This reversion happens somewhere else in the genome, and the strain becomes neurovirulent once again. Just again, I'm repeating that. So the P3 Sabin, you have the 472 C2U mutation, and it becomes attenuated. But it has been observed that that mutation remains, but somewhere else, naturally, as the vaccine is given to children and others, a mutation somewhere else in the RNA can revert the P3 strain into the virulent strain. And that causes a large number, not a large number, like considerable number of vaccine, polio out of the vaccine. And these are some of the things that you have the live vaccine problems, and these are the things what people fear, that live vaccines can get the disease. Yes, so due to the polios that we see now, they are in small pockets of Afghanistan and in a few pockets of Africa. That is the only 2 pockets that the polio is still there. And the polio, what is existing in that, they are all from the vaccine strains, unfortunately. So that's what it is. It's diminishing, and hopefully in a few years it will be eradicated, but they are from vaccine strains. All right. So here we are looking at this 2 different viruses at the 472 mutation of the P3 Sabin strain, and you can look at that. Now remember that the panel A and B is entirely different. So in the panel A, we are looking at 2 strains of this virus, and you have a base change at the 472, you have an U, and you have a C. So you can see that LD50, or the lethal dose 50, where 50% of the mice will die, it is 20 million. If you just let lethal dose, you have to give 20 million virus inoculation so that you get a 50% mice will die. Whereas if you have the mutation of C instead of U at the 472, you can see it's about how much? 9,000. So that's the difference, 9,000 versus 20 million. So you can just easily imagine that how different in the LD50 by the number of virus particles. So here, on the other hand, what you are looking at, that this is the log 10 of the PFU and the number of post-infection, and this is the replication rate of the virus. So you can see that the virus is still replicating pretty well even after 2 weeks. But you can see that the viruses with the mutation with the EU, they are drastically dropping. So see that there is a difference in there. So this one right here with the C, the LD50 is less. The LD50 value is less, but right here you're looking at that this is replicating. So remember that always the LD50 value. So out of those 2, the PRV 7.3 and 8.4, which is more virulent? 7.3 or 8.4? Think about that. LD50, one is 20 million for the 7.3, and you have 9,000 for the 8.4. No? Think about for your microbiology. Less the number, more virulent or less virulent? More the number, less virulent or more virulent? Just think about this way. 20 million virus particle, you have an LD50. You have to give more virus to get the people die or the mouse die. In the other case, you are giving the less amount of virus for the mouse to die. So what happens, the lesser the number, the virulence is more. Higher the number, the virulence is less. So the first one, which is EU, that is less virulent, and the C is the more virulent. And here you can see that this actually shows you that why it is the C with the EU virulence right here, you can see the viral replication drastically drops, whereas with the C, which is much more virulent, you have a constant production of the viruses over the period of time, days of post-infection. So just remember that, and that's why I say carefully understand the rationale of the experiment. Just one thing, always remember that LD50 value, the number, if less, more virulent. If the number is more or higher, it is always, if the number is higher, it is less virulent. If the number is less, it is more virulent. It's a reciprocal relationship. Just the way it is, think about that you will kill 10 mice if you give them say 10 viruses. You kill 10 mice. in the, or not 10 mice, the LD50 is 50% of mouse will die. On the other hand, you need to put 1,000 viruses to kill the same number. So that means that the 10, when you just need only 10 viruses, they are more virulent compared to the 1,000. That's the rationale. So just remember that. All right. So here what we are looking at, the gene products that modify the host defense. This is the next category that we are seeing. So the first category that we've seen, the replication. So here, replication. We are next going into the modify of the host defense mechanism. So these produce, we have already seen that they can change the apoptosis or the autophagy. And also there are some intrinsic proteins that is counteracted. Say for an example, the HIV, ApoBec3G is the, remember that. I just wrote together, but just remember that HIV doesn't have the ApoBec3G. ApoBec3G is a host protein. It is in us. The VPU is the HIV1 protein, which basically forms an ubiquitin ligase around the ApoBec3G to degrade that and counteract. The same way that another intrinsic protein, the HSV ICP34.5, that is a virulent gene, and if you knock out that gene, the HSV infection just doesn't stay. So very important for that. And many of the viruses do produce virokines or the viroceptors, and these are basically the mimicry molecule. They look like chemokines or cytokines. They look like the receptors. These are the typical ones. And so basically what it is doing. So just think about that you have a cytokine receptor, say for an example an interferon receptor, and the virus is producing a molecule which mimics the interferon and binds to the interferon receptor but will not cause any signaling. So it's blocking the receptors. On the other hand, the viroceptors is the way that these are soluble viroceptors. That means that they are not on the membrane. The virus is just producing them as soluble, and you have an IFN, say IFN1, and you have a viroceptor for IFN1 in a soluble condition. Cells secrete the IFN, it just goes and binds them and basically use it as a sink. So it just drains out these virokines out of those viroreceptors. So this is how it works. And then you have complement binding protein. We talked about the smallpox spice. So those complement binding proteins, modifier of MHC1-2 pathways, we have seen that as well. HLA-E overproduction, we have seen that. And so these are the host defenses. And often these are not required in the growth in cell culture, because remember that a cell culture is a cell culture. It does not have an immune system. The cells can produce interferon when infected, but out of that, where it will get B cells, where it will get the dendritic cells, where it will get macrophage or the T cells, doesn't have any immune system. All right, so here we are looking at viral virulent genes, and carefully also understand that experiment, that what happens. Here, gamma herpes virus 68, and the M3 gene encodes for a chemokine receptor. And what we are looking at is the dose PFU right here in the bottom x-axis, and right here percentage of survival. So the blue plot is the wild type. So you just infect with this dose, and you can see that right here. Look at the 50% survival. That will be your LD50. Remember that. We are infecting the mouse with different doses. Those are these points, different doses, and we are just looking at a dose response curve. So if we look at the 50, that is important. This is your LD50. You just go straight and see that right here it intersects with a blue line. And if you just go straight down, approximately, just like eyeball, you say about 5. So the 5 PFU is the 50% LD50. So for the LD50 for the wild type virus is about 5. Now see what happens, that you delete that M3 gene, which is responsible for the chemokine receptor, and now look at the LD50. So I'm going all the way down right here. Right here, this is the point which is intersecting with the 50. And if we just see, it's like what, 300, 400, something like that. So now you need 300 or 400 viral particle to cause 50% mortality. Here, it was just 5. Here it is 300, 400, something like that. Now what you see that this is the MR, that means that this is, you initially delete the M3 gene, and MR is a marker rescue. That means you supplement back or give back the M3 gene to the virus again. Marker rescue is an old molecular biology term, but just remember that you have given back the gene to the virus of the M3 which you deleted initially, and see that it just goes back to exactly the wild type level. So you can just determine that this is the virulence gene by these experiments. All right, so after that we are looking at all of these. Modify host defense, enable virus to press to the host, and then now you have intrinsic cell killing. So here is an example of a toxic viral protein which can basically kill the cell. Here the rotavirus NSP4 is a non-structural glycoprotein of the rotavirus. It's not a structural part of the protein, but it is an enterotoxin. So what it does, that just remember that sodium or the glucose, it just blocks the sodium glucose luminal transporters, basically. So when you stop this, so basically what it does, it just sodium goes in and it just takes glucose, so it's a co-transporter. So as sodium goes into the cell, remember that just to maintain the electroneutrality, the chloride ion, more plus goes in, so it has to maintain the electroneutrality. If the cell will reabsorb chloride, Cl minus, Na plus goes out, it will need the Cl minus inside the cell to maintain electroneutrality. So if it blocks the sodium glucose luminal transporter, that sodium chloride is in the intestine. And remember that salt attracts water. When you have that much more salt in the intestine, that's going to take out the water from the cell, cause the diarrhea. That's number one. Also, along with that, NSP4 basically activates another signaling molecule, which is the phospholipase C-based signaling molecule, PLC. So the PLC actually signals through a calcium-dependent chloride transport. So what happens, this calcium-related chloride transport secretes more chloride into the intestine. So see that first of all, sodium glucose luminal transporter stopping sodium to come in. That means chloride is not going to come inside the cell. On top of that, you are releasing more chloride through the calcium-dependent manner into the gut. That will draw more water out of these cells, and that causes the diarrhea. So direct toxic effect of the viruses. So here what we are looking at, that cell injury, this is basically the cell killing process, and cytolytic viruses shows the cytopathic effect like apoptosis, necrosis, pyroptosis. So the difference between these differences are, so the apoptosis is your programmed cell death, that there are some specific way that caspase is mediated or where the cell commits the suicide. Necrosis is not programmed, is basically killing the cell directly. The cell doesn't kill itself. The cytopathic effect of the virus kills itself. And the pyroptosis is basically, as you can see, that think about that fever is called, that is causing by the pyrogen. Pyro means heat. So the pyroptosis is basically the immune-mediated killing of the cell. So your immune reaction, cytokine, chemokine, which can kill the cell. So you will ask that why it is, how it does. So many of the cytokine and the chemokine will release proteases. They release superoxide radicals like ROS, reactive oxygen species, O2 minuses, and like TNF alpha, TMA necrosis factor alpha. These are all cell-killing machinery, and your immune system can kill the cells. They try to do it to kill the viral infected cells, but sometimes it will cause tissue damage to the healthy cells as well. Viroporins, they can just make pores through the plasma membranes, like influenza M2, picornavirus 2B, HCV-P7. They all make holes through the plasma membrane, viroporins. Remember that aquaporins are the channels through which the water is transported through the plasma membrane. The same is viroporin, are basically the channel through which it just makes the plasma membrane porous and make holes through the virus, holes through the membrane to lyse them. And we have seen this, doesn't need to recap. Basically, host protein RNA synthesis leads to loss of membrane integrity, leakage of enzymes from the lysosome, cytoplasmic degradation. We talked about a lot about the protein and RNA synthesis inhibition, how it is done. And also, you can see that there are some syncytium formation, which is the fusion of cells into these large giant cells. Here you see 1, 2, and 3 of these. Remember that these are not syncytium. These are the clump of cells, which does have a bright border. What I'm pointing out are these giant cells. So the ones right here, with these are the nucleus. So see that 1, 2, 3, 4 nucleuses probably. These are the giant cells, which doesn't have that bright border. Those are the giant cells, or the syncytium. These are the cytopathic effects. So immunopathology is that we are going to talk about here now is too much of the of good thing that can kill you. And you can easily see that we already talked about that fever, tissue damage, aches, pains, nausea. These are all consequences of response to the infection. And the non-cytopathic viruses, the disease is usually the consequence of immune response. So just remember that. These are the effect of some of the viruses which directly have a cytopathic effect. But all viruses don't have a cytopathic effect. They don't kill the cells directly. But in these cases, sometimes your immune system kills you or kills your own cells. And that's basically the problem. So here, some of the very, very well-known immunopathology, the number one probably comes to your mind is the cytokine of SARS-CoV-2. So the people who are dying basically in the ICU and all those things, the cytokine storm is the main problem. Too much delayed cytokine storm, and we're going to talk about that. That is then CD8 T cell mediated. Remember that the CD8, these are the cytotoxic T cells. They can kill your normal cells as well. This is the CD4 T helper cell mediated, Th1 in measles virus, or the CD4 T helper cells, type 2 respiratory syncytial virus. And also they can be B cell or antibody mediated in the dengue virus. We're going to take a look at a couple of these in detail. So let's take a look at this. So here what is happening, that this is in the SARS, what has been shown, that if you have an early interferon signaling, you have a reduced virus titer, regulated inflammation, and a mild clinical disease. So in the patients that that interferon is given to them or injected to them at the very onset of the disease, at the very beginning of that, they may not have a full manifestation of the disease. On the other hand, if there is patients who are there is no IFN signaling, and they do have a weak immune system. Now remember that, or suppressive immune system, they may not have a disease as well. On the other hand, what happens that in the delayed IFN signaling, where you do not have see that the SARS-CoV-2 titer goes very, very high, and when you have a delayed IFN response, it actually shows the dysregulated response, lung immunopathology, lethal pneumonia. This is basically the cytokine storm, which is often lethal. So this is so think about that. Early IFN interferon, great. If you do not have an IFN, still you can probably survive, but the delayed IFN is sometimes could be lethal. So here you do not have to memorize or do anything in here. Just showing, this is a paper from Lancet in 2020. So what we are looking at, that just the red boxes are the inflammatory cytokines. And you do not have to remember any names or anything. I just wanted to show two things in here. So if you have a darker red arrow, this is your basal cytokine level. That if you have in a non-infectious condition, that this is your basal cytokine level. And they measured the ICU patient's cytokine level about SARS-CoV-2 infection. And I just want you to just look at the basal darker versus one. So just look at that how elevated the cytokine level is. This is the basal level, higher level. Basal, higher. Basal right here, up to this. So all of them, that what is being shown, that in ICU patient, this huge amount of increase in the cytokine, which are the inflammatory cytokines, this is basically the killer. This causes the immunopathology, and we're going to see that what these basically cause. And I just pointed out these, but the other ones, some of them pro-inflammatory cytokines, chemokines, everything is elevated. So it is kind of like mind-boggling that what this virus can do. And this is in SARS-CoV-2, remember that. This particular example, I probably should have written somewhere. So right here, what it does. Healthy, infected, moderate damage, and severe damage. So if you are looking at this ARDS, acute respiratory distress syndrome, in a moderate lung, that what we are looking at, this is a single alveoli for gaseous exchange. And right here is the capillary around that through which the gaseous exchange should happen. And each alveoli is lined by two types of pneumocytes. And again, this is not detailed, but just if you're just interested, you have a type 1 pneumocyte. 95% of the alveoli is type 1 pneumocyte. They are the thinnest one. These are for the gaseous exchange. About 5% are the type 2 pneumocytes. These are the cuboidal shaped pneumocytes, about 5%. They are granular. They produce the surfactants. So type 2 produces surfactants, and the surfactants is essential for the gaseous exchange. If you do not have the surface tension reduced in between the empty space where you have the gas, oxygen or carbon dioxide, and on the plasma membrane, gaseous exchange will be drastically reduced. So that's the reason that what you have. So here you can see that the flat cells are the type 1s, and this is the type 2. And when they're infected, they are going to produce the inflammatory signals. Macrophages will come in that you can see that they invade the lung through the diapedesis or squeezing through these capillaries, from these capillaries and the pneumocytes. And you have the pulmonary edema. You will have fluid discharge into your lung, and you don't have the half of the alveoli for the exchange of the gases. You still have some, but not half of the lung. Moderate damage. Here what happens, on the other hand, what you are seeing in extreme cases, you have formation of these scar tissues. Here, basically, the pneumocytes are killed, and that space is basically produced on a hyaline or a cartilaginous membrane from the connective tissue around them. You have the killed cells produce this protein and cellular debris. Neutrophil comes in to phagocytose those, and at this time there is so much of fluid that basically the whole alveoli basically drown in the fluid, and there is basically no gaseous exchange. This is basically the immunopathology. Completely, virus is not doing anything, just the infection caused this, or your own cells. So here is another way that just shows the blue portion is the surfactants along with the type 2 and 1 pneumocytes in a cross section, and these are the inflammatory cytokines. Tumor necrosis factor, IL-6, IL-1 beta, CCL2. These are the immune mediators which basically cause this immunopathology, killing the cells. This is the hyaline membrane, which is basically a scar tissue. If you have this hyaline membrane formed, here there will be no gaseous exchange through this. And you can see that almost there you have a pulmonary edema, which is the fluid. And you can see that right here you have healthy amount of surfactant, reduced amount of surfactant, almost no surfactant, almost no blue. And these are the tissue damages that is caused by the SARS-CoV-2 by the inflammatory cytokines. Here we are looking at an example of CD8 T cells mediated. So here the lymphocytic choriomeningitis virus, if you just inject them to the mouse cranium, you have a lethal choriomeningitis and the mouse is dead within a week, 8 days. Right here, if you inject it to the mice, in an immunosuppressed mice, you have a persistent infection, but it survives. Think about that. Without immune system, it survives. You inject them with the CD8 T cells back again, it just causes a lethal choriomeningitis and it dies. So here directly the cytotoxic T cells are the ones which is responsible for the lethality. That's the unfortunate part. Too much of a good thing but it can kill yourself. So here, what we are looking at, lesion associated with, on the other hand, CD4 lymphocytes. These are the T helper cells, remember that, unlike the CD8 cells, which are the cytotoxic. CTL, cytotoxic, CD4, helper. They have more elaborate cytokines than the CD8 cells. Remember that, because they are the helper ones which release the cytokines to recruit the T cells or the cytotoxic T cells as well as the B cells. So they are more important important repertoire of cytokines and activate many non-specific effector cells. Most recruited cells are the neutrophil mononuclear cells, which are protective but cause tissue damage, as we just saw. Immunopathology caused by release of proteases, we were talking about, reactive radicals, reactive oxygen species mainly, and the cytokines like TNF alpha. These are all basically can damage our own tissues. So here is also examples of some of the CD4 or the CD4 T helper cells related immunopathology. This is in the herpes stromal keratitis. This is actually the #1 common cause of blindness in developed countries. Just remember that. Because, as I said, like we are all have some herpes virus somewhere in our body, mostly in the latent stage. But this is entirely by the Th1 cells, and repeated infection cause opacity and the reduced vision, and basically the cornea becomes opaque, or as you can see, it just become white. So how this happens? On the left-hand side, we're just looking at the cross-section of the cornea, epitheliums, then you have the stroma, and then you have endothelium, which is a dermis, basically, normal. Here, what we are looking at, that the virus replicates in the corneal epithelium, but the inflammation is restricted to underlying stromal cells. So in this one, what you are seeing, the green ones are the areas where the virus basically replicates in these cells. So you can just see that these are the lesions that are formed. But this is not the main cause of the blindness. What happens in the last stage, the stromal cells, which is the stroma is this right here, is the damage by the secreted cytokines produced by the infected cells in the corneal epithelia. So what happens, that these are the infected cells. These are going to release the cytokines, and they are going to attract all the CD4 cells. So right here, again, not very clear, but just remember that. These are the CD4 T cells. That's pathologically, you can determine those under the microscope. Here is almost impossible. These are the CD4 cells, and they all home in below the cornea. So basically, again, remember that what I'm trying to say, that these are the corneal cells, epithelial cells. They are releasing the cytokines, and they are going to attract the CD4 T cells under the corneum. These CD4 T cells are going to secrete the cytokines, which will cause the tissue damage or the corneal damage, which will lead to the opacity of the cornea to blindness. So here is also another example, that TLR3. If you have, and how it is in the West Nile encephalitis, that how it is implicated. So here you were seeing that TLR3 minus means like it's completely TLR3 is deleted. The virus is more resistant to West Nile virus, impaired cytokine production. So what is looking at that in a wild type, see that day 0, day 3, and this is just to look at the blood-brain barrier. You inject the virus with the Evans blue. It's a dye. So it tells you that whether the blood-brain barrier has been compromised or not. So here you are looking at that right here. The brain has taken up the dye. That means that the blood-brain barrier has been compromised. So you can take it up. On the other hand, you can see that TLR3 mice right here, the blood-brain barrier is not compromised when you have the deletion of the TLR3. So basically what happens in the wild type, after infection, due to the TLR3 response, TNF alpha is produced. This causes the blood-brain barrier breach, and the virus can reach the brain and kill the mice. We have seen that. On the other, or cause the encephalitis. You can see inflamed swollen brain. On the other hand, you can see that if you just delete the TLR, it takes much more longer time before the blood-brain barrier is breached and not at this level. So these are some of the responsibility. Then we are looking at some of the poxes and rashes. Many viruses produce characteristic rashes. We have seen that. Smallpox, varicella zoster, Th1 macrophage activated and homing onto the foci in the skin. And also the cells produce these IL-2, IFN1s. And these local increase in capillary permeability and T cells produce these rashes. This is very straightforward as well. But the important part is that we are also looking at that the dengue, which is probably called the breakbone fever and probably just most important after malaria. Let's take a look at that, what happens in here. Usually, and the descriptive part I'm not describing, it's just read it one time, the descriptive part is good. So causes a severe febrile illness, and those part I'm not talking about, just talking about that what the important part. 1 in 14,000 in primary infections have the dengue hemorrhagic fever, which is life-threatening. For most of the people, they recover within 7 to 10 days. So 1 in 14,000. But what happens that there are 4 serotypes. So dengue type 1, type 2, type 3, type 4. They can develop these antibodies against these serotypes, but offers no protection. What it means, you have dengue 1. After that, you can be reinfected by dengue 2 or reinfected by dengue 3, dengue 4. They will not cross-protect. If you have dengue 1, you may not have dengue 1, but you can have the other ones. So here is the problem. So these monocytes, what they have, that they have these Fc gamma receptor, which binds to the antibodies. These monocytes mature into macrophages, remember that. And they can internalize the virus bound to the antibodies, basically. So you have the dengue virus, and you have these heterotypic antibody from the primary infection, and they remain. In the secondary infection, although there is no cross-reactivity or protection, remember that the antibodies that is produced by the one type of infection cannot stop the disease, but some of these antibodies can recognize the other. So remember that it is not neutralizing, but can recognize to some extent some of the viruses of the other type. When they are bound to those viruses, they bind to these Fc gamma receptor of the monocyte, and they infect the monocytes. So the monocytes now become an additional place for the replication of the virus and increases the viral load significantly. So this is an antibody-mediated secondary dengue infection or tertiary dengue infection. And see what happens. In a secondary infection, the chances of the hemorrhagic fever goes up to 1 to 90, 1 in 90, or in the tertiary, 1 in 50, from 1 to 14,000. So this is the antibody-mediated enhancement of disease. And then we have this global suppression of the disease in many different ways. Replication in one or more cells of the immune system, cytokine homeostasis perturbations, intracellular signaling. We have all talked about these, viroreceptor, virokine production. And this is by the measles infection. So this is a tuberculin test, and by the tuberculin test, by measuring that how much of a rash that you develop, or the skin rash that you develop after that determines whether you have a TB or not. But see that right here, what happens with the measles virus. So this is the diameter of the rash, basically, that what you have in centimeters. Say, for example, you have a 6-7 centimeters of the rash. But after the measles, the onset of the measles, you have almost no tuberculin test. There is it's just like completely immunosuppressive. Then after about 2, 3, 4 weeks, it takes about 4 weeks by which your immune system will recognize that you have a TB test. So this is purely immune suppression during the measles infection. And we have seen this before. So what it does, that see it binds to the TLR4, and these antigen-presenting cells produce this IL-12 to activate the CD4 T cells, and these are the Th1s. And they basically activate the CD8 T cells. This is the first thing that should happen to infect the cells which are replicating the virus. But what happens, the measles virus skews it back to the Th2 type. We already talked about that, because they can produce these antagonists, which is IL-10, which basically suppress the IL-12 function, and it skews it back towards the CD4 T cells of the Th2. This favors antibody production, but initially this is not that is required. So basically this is the Th1 response is required in the first instance, but once it skews it to the other side, this is what is caused, that basically the immune suppression is done by the CD8 T cells like this. So here, these are some of the examples: monocyte DC thymic epithelial cells, reduced T cells, enhanced infection, rubella, German measles, lymphoid cells, persistent rubella infection, HIV. It directly infects the CD4 T cells and the monocytes. And in that case, opportunistic infections or the neoplasia, which is the abnormal growth in many tissues or cells, can occur. So that's pretty much it. Where in the world we finished up 3 minutes before. So this was like a pretty much any questions so far? Good. All right. So I will make your PowerPoint slide for the for the exam with those star dotted ones, and I hope that by tomorrow evening I will be able to upload that, and then we'll have Thursday we'll have the review, and you have this time, you have plenty of time. So what I highly suggest that just go through the slide and watch, and if needed, rewatch the review. Like, you stop at one time, take notes on that slide, that what is being said and what could be in there, and it will be very thorough. If you do your homework, that last time you didn't have much time. Well, you will have, but you have a break and all those kind of things. But here you will have more than 1 week with the material, so you should be able to do well. And then Tuesday we'll just add the honor slides in there, and that's about it.